Good morning, everyone. The next talk, the next speaker, is Dr. Luca Pozzi. Dr. Pozzi is an assistant professor at the University of Texas at San Antonio. Dr. Pozzi research has focused on understanding species diversity in morphologically cryptic primates species, using a multidisciplinary approach that draws from phylogenetics, population genetics and genomics, biogeography, and behavioral ecology. His talk is titled Hidden in the Dark: The Importance of Integrative Taxonomy and Museum Collections in Exploring Cryptic Biodiversity. So, Dr. Pozzi, you may start whenever you're ready.
Okay, let me just share my screen. Okay, hello everybody. Thank you so much. First of all, I want to thank the organizer for inviting me to this conference, and most of all to actually organize this conference. I think this is has been a great idea, and I hope this is going to continue after the end of the pandemic, so that we can spread science and our research to more people, and also not just people that can participate in big conferences. Before I get started with my talk, I also want to acknowledge that the work that I'm presenting today and all the work that I've been doing in my career is the outcome of collaboration, as very often is in science, with a lot of different people, including a lot of amazing students, undergraduate and graduate students, and also colleagues in the US and in many other area of the world. I have a big, wide international network of collaboration. And also, of course, I need to acknowledge the funding that made this research possible, including my home institution, UTSA, the American Museum of Natural History in New York, where I have a strong collaboration with, and then, of course, the National Science Foundation. So today I'm going to talk about cryptic diversity, and in particular, how we can define species within cryptic diversity. As you probably know, describing diversity is, by definition, one of the most important tasks in evolutionary biology. We are all interested in understanding the processes and patterns of generating biodiversity. And so understanding species diversity in specifics is critical for a lot of different tasks, not only for just a theoretical question, but also to really use it as a unit to study ecology, evolution, behavior, and of course, in conservation. And being a primatologist, I'm also very interested in conservation, and understanding what the unit of conservation is critical. So we need really to define what a species is. But as you probably all of you know very well, the species problem is arguably one of the most contentious topics in evolutionary biology. And so there's been a lot of debate in defining what a species is and how we can delimit them. So there've been a lot of different concepts of species that have been proposed over the years, starting from the biological species concept proposed by Meyer and Wright in the 40s. And here is just like a table from a paper that now is almost 15 years old by the Kerouacs, showing how like there's so many different species concepts, different variation of the same species concept. They also have different properties and different criteria that they are used. So this aspect is not only at the theoretical level, in which we are really defining what a species is, what the concept of species is, but is also a practical one, because different species concepts also include different criteria that we can use to delimit those species and use in practice to define speciation. And of course, the concept of species is tied very closely to the process of speciation. So if you're looking at this graph, on one side on the left, we have a situation that is pretty straightforward, right? So we have a panmatic population, all the individuals are like mixing with each other. And so very easy, we can define that as a single species. On the other end of the spectrum, we have a clear, full, reproductive, isolated population. And so those two entities can easily be called species A and species B. The main problem that we have is in the middle during the speciation process, in which at different level, we might have this so-called gray zone of speciation, in which some criteria might define it as one species, and some other might define it as two different species, depending on what kind of tools you're using to define a species. In fact, when we're looking at the gray zone of speciation, different phylogenetic concepts or different species concept might use different biological evidence, different lines of biological evidence. And these different lines of biological evidence might provide a different answer to the same question. That is, how many species we have out there. So as we'll see later, this is not necessarily a wrong and a right answer, but it's simply because different aspects of the biology of a particular organism, their behavior, their ecology, their genetics, might change at different time during the speciation process. So what I'm interested in and what my research has been mostly about for the last 10 years or so has been about cryptic speciation and cryptic species. And when I talk about cryptic species, I talk about morphologically cryptic species, means basically animals, organisms that have very subtle, if any, morphological variation. So as you can see here in this slide, this is one of my favorite group that I studied as a postdoc when I was in Germany. This is the mouse lemurs. As you can see here, the morphological variation within the genus Microcebus is very limited. So all the species at first look very similar to each other. However, the diversity that we have in this group is enormous. And we have over 20, 25 different species of mouse lemur that are very similar to each other at the morphological level. So the major question is, how can we define cryptic species? How can we draw a line in the species boundaries when it's so difficult to define the morphological variation that we have in these animals? So as you know, the use of molecular data, genetic data, started from the late 80s and the 90s, and of course, more recently with genomic information, has really led to an increase in cryptic species. This has been in all different taxa, but of course, in particular in cryptic taxa, like mammals and nocturnal mammals, including primates, in which we saw recognition that the diversity that was known before the advent of molecular data was much lower, was much higher, sorry, than the one it's actually existing. So the question here is, how is our ability to use molecular data to define cryptic species is biological meaningful? In other words, if we're using a segment of the mitochondrial DNA or any other piece of our genome, of the genome, to define different species, is this biological meaningful? Or is just an artifact in the way that we are defining different species? So I would argue here, and this has been the main focus of my research, that we really need to apply an integrative approach to identify biodiversity. And when I talk about integrated taxonomy, it's basically the idea of combining multiple and complementary perspectives about the biology of a particular organism in order to define a species. So this includes phylogeography or biogeography in general, morphology, obviously, and genetics, but also ecology, development, and behavior. Because all these different evidence, different lines of evidence can provide us not only a much more clear information about the lines, the boundaries between different species, but also can shed, as we'll see in a little bit, shed light on the process of speciation itself. So the fact that different lines of evidence are against each other or they don't agree with each other is not something that we should see as a bad thing, but we should actually see it as a way to have a better understanding of the speciation process, as I will show in a little bit. And here brings me to my study group. So I've been working on nocturnal galagids for quite a long time. And this is a very interesting group of primates because this is probably one of the most cryptic family of primates out there. We currently have 20 plus species. As I said, diversity is probably strongly underestimated in this group, divided in 6 different genera. So the fact that this group has been poorly studied is mainly due to the fact that they are highly cryptic, so very difficult to study in the wild and very difficult to recognize and tell apart as different species. But there's been a lot of different aspects that also slow down in a way the understanding of their biology and diversity. And this is very common in a lot of the taxa that we're studying. Of course, we all would like to have full genomes and working with genomic resources, but this is very difficult for many taxa, including some of these primates. We had historically a lack of high quality sample for this group. These animals are extremely difficult to capture, so we lack genomic resources. It's very difficult to access a wild population, mainly because not only they're hard to sample, but because they have very widespread distribution, and they very often live in areas where it's very difficult to work for social political issues. And also because they're primates, they are constrained by a lot of regulation in collecting biological samples. And so compared to, for instance, studying cryptic rodents, very often we are not allowed to sample them, we are not allowed to trap them or to collect biological samples. And so all these aspects of their biology and the logistic made very difficult to understand these primates. However, this group is extremely interesting because it represents a very good case study for one of the species concept that has been proposed. And this idea is that, as you can see in this slide of some of the species that are present in the family, they're characterized by very specific vocal advertisements. That means that they have very specific calls that they use, and advertisement calls that they use in particular for recognizing member of the same species and for mating. And so they've been suggested as a possible indicator of species identity. So even if species are very similar to each other, so they're cryptic at the morphological level, having different calls might allow them to identify as different member of the same species. And as I said, these perfectly follow one concept, species concept that has been proposed, that is a sort of a evolution, if you want, of the biological species concept, that is the recognition species concept, that suggests that basically species are defined as group of organisms that share a common specific mate recognition system. So in other words, there are species-specific signals. They play a critical role in species recognition. And so if those calls, for instance, or those signals are different from each other, they might restrict gene flow between different species. And so the example of the Galagos is actually a very good one, because it's possible that those calls, those acoustic signals, might be used as a way to restrict gene flow between different species. And this is where the integrative approach comes very handy, because we can study different aspects of their biology, their behavior, the bioacoustic behavior, their ecology, genetics, and morphology to really tease apart how these species recognize each other and how they can restrict gene flow between members of different species. And so that's what we did. And one of the things that show how poorly known this group is that just a few years ago, by studying and using an integrative approach across the family, so combining bioacoustic analysis, biogeography, morphological analysis, and genetic, we identified a full new genus in this species, in this group, in this family, the genus Paragalago. And this is quite exciting, not only because, well, you know, describing a new genus, if you work in taxonomy and phylogenetic, is always kind of exciting, but it's also because it really shed light on the processes of evolution in this group. In fact, it told us that there were two groups that were very similar to each other, what we call the dwarf Galagos, that they were not even related to each other. and they were extremely different in so many aspects of their biology, suggesting that we see a sort of convergent evolution in their body size. That is extremely interesting because now we can study the processes of this convergent evolution and try to understand why this happened. What are the forces, the drivers that are leading towards this body size? And so, again, this is an example in which the integrating different evidence not only allows us to clarify the taxonomy, that of course is very, very important, but also allowed us to understand better the patterns and processes of diversification. And here is another example that I've been working on for quite a long time, that is the so-called ZANZIBARICUS-COMPLEX. This is a group of probably 3 different species. As you can see here, these are 3 different individuals of 3 different acoustic species. And when I talk about acoustic species, I talk about species that have been defined based on their acoustic repertoire. And so as you can see them, they're almost identical to each other. The level of variation that you find them at the external and internal morphology is minimal, if any. However, they show very clear different advertisement goals. And so they've been defined as different species, but the evidence to test this hypothesis has been very poor until recently. So what we did is to use, again, an integrated taxonomy approach in which we use both single locus mitochondrial DNA and multi-locus data from multiple loci, nuclear loci. And we use different methodology. As you're probably aware, there are a lot of different species delimitation approaches out there. And we use them to compare them against each other to see what genetics was telling us about their diversity. We also did analysis using the bioacoustics, also running playback experiments to see the ability to recognize members of a similar species or a different species. And we ended up finding 3 clear clades or three different groups that were totally different from each other. And they actually ended up diversifying very early in time, around 3-4 million years ago. So again, this allowed us really to identify species diversity in this highly cryptic group. But even more interestingly, it gave us something to think about that goes beyond species boundary, as I mentioned. Because we were able to combine different evidence from different aspects and different components of their biology, we are now thinking that their speciation process happened through a process that is called sensory drive. And it's basically this idea that signals can change due to their environment. So if you follow this graph here, we see that starting from a signal that can be used for mate recognition, this signal might adapt in different ways depending on the habitat. For instance, a more closed habitat like a forest habitat or a more open habitat. And this is barely just because of the ability of the sound to propagate in space. But also it can be affected by the background context. That is basically this idea that different sounds, like background sounds, like other insects, other animals, or simply like climatic aspects like the wind and so on, may create a different interference with the signals. And because of that, the sensory system of both the emitters and the receiver adapt its tuning in this particular condition, and is driving 2 different signals in 2 different populations. And at that point, those 2 populations become completely isolated because they simply can't recognize each other as members of the same species. So the sensory drive is a very interesting hypothesis that now we can test using different methodologies. So in fact, we are now able to combine demographic models and spatial analysis to really identify how this sensory drive might actually be working. And again, this is the idea that combining multiple hypotheses or multiple lines of evidence, can really provide information not only on the taxonomy, but also on the speciation process. And finally, I want to stress one of the most important aspects for studying cryptic speciation, that is the ability to collect samples in museums. So this is a project that I've been working for a couple of years together with Dr. Mary Blair at the Museum of Natural History in New York and sponsored by the NSF. And this allows us to collect genetic data, finally genomic data. We are bringing the GALAGIDS to the genomic era as well, from museum specimens. And this gave us like two major advantages. First of all, it allowed us to include more species than we ever had before, simply because, as I mentioned, a lot of these species are very difficult to capture in the wild. And so now we are able to get a genomic data set of 334 loci and also full genomes that we are in the process of sequencing of over 100 strepsirrhine primates that include the Galagos and their sister group, the lorises and the lemurs, and allowed us to really resolve some phylogenetic questions that we'll not be able to solve with limited genetic data. But more interestingly, for the cryptid diversity question, the benefits of the museum collection are related to many different factors. First of all, we have a lot of species that we can access, and in particular specimen for the same species. We literally have, as you can see, are drawers of specimen that we can collect. They have very often very good geographic location, so we are able to get a diversity of geographic diversity, and so allowed us to have a better idea of their within and between species diversity. And finally, we can combine genetic data collected by the specimen with morphological data from the same specimen, because we can study them directly on museum. And this can really bring us to the next level to study cryptic diversity. So with this, I want to thank again the organizer for inviting me and for organizing the conference, and my institution, UTSA, the American Museum of Natural History, and all of you for being part of this seminar. Thank you.
Thank you very much, Dr. Pozzi, for your talk. I'd like to remember everybody that you can write questions in the chat below, and I will read them for Dr. Pozzi. So we have two questions now. The first one is from Laisla Cagliari Costa. She asked, how different species concepts can affect this research?
They can a lot. As I mentioned, if you are based your research on a single species concept, like, a lot of the research, for instance, the early research, for instance, on lemurs, was done a lot of with the phylogenetic species concept. And so just by defining or identifying single differences in mitochondrial DNA, they were describing them as different species. But if you're using them a different system, a different species concept, you might not define them as different species. So that's why I argue that an integrative approach is more relevant, and not just because it's hard to draw the line, but it's because it really gives us more information about what's going on there. If we have mitochondrial differences, what does it really mean for the species? Because at the end of the day, we need also to think about the species perspective. So how is the species really recognize members of the same species? So there's clearly definitely, the species concept can definitely affect this research in many ways.
Awesome, thanks. Laisla asked another question. She asked if there is evidence on how urban development can affect the bioacoustics of near forests. May urban development near protected or natural areas influence any insensory drive and later in speciation?
That's a very interesting question. Yeah, we expect that animals that are really driven by acoustic might actually be affected in a way in their biology. Of course, in urban area that might be very, very loud. And I think there's been some very good studies done in insects like Orthoptera, like crickets and grasshoppers, that they communicate with sounds, and birds as well. Yeah, and also, if you go in a different sensory spectrum, even for lights, animals that, communicate with lights, bioluminescence, for instance, they've been affected by urban development quite a lot. I think this really in terms of affecting speciation, depending on the taxa that we are studying, we need a lot of generations to really see that happening, right? So, you wouldn't see that on primates. It's likely that primates go extinct in urban area much faster than even having any changes in their evolution. But you could actually maybe see them in animals that have a faster generation time, like in insects, for instance. We might see these differences. And we have some interesting, and this is not related necessarily to sensory drive, but a lot of field of urban ecology and urban genomics that is really studying how different species are adapting to urban environment. So there is a lot of good study about how rodents, for instance, in New York City have been adapting to metabolize heavy elements, for instance, because they're more subjected to heavy elements in the city compared to like more rural area. So this is a very interesting study. I don't know exactly if there's anything that has been done on speciation specifically, but it would be very interesting to understand that, yeah.
We have one last question from Valeria Gallo. She said, thank you very much for your wonderful talk. I wish to ask you about the restriction of the concept of subspecies or historical sublineages. I did that because she
yeah. Thank you, Valeria. Yeah, that's also a great question. And, if the species concept is kind of controversial in a way, the subspecies concept is even more controversial. And, I think there is less theoretical framework for subspecies, and people have been describing subspecies in different way as morphological variants or behavioral variants of a species, or as incipient species. So, group or population, they are sort of diverging, and then maybe eventually they will become different species. I think, again, I'm actually working, and I didn't have time to present it, but on a different group that is the same distribution of the ZANZIBARICUS-COMPLEX, in which we are redefining subspecies, and we are sort of looking at what kind of criteria we can use in that sense. And I think, to me, again, I would use the term subspecies when there are some morphological diagnostic feature, but you don't see enough evidence to see a full reproductive isolation between those groups. And so, for instance, genetic data might not be giving you nice, clear monophyletic group as you would like to see in species definition.
We have one more question, and that will be the last question because we've blown our time limit. I'm sorry, I will most likely butcher your name pronunciation. Mumin Michael Mentin asked, what are the characters to consider in cryptic species? Does morphological traits or genes contribute to its identity?
Yeah, it really depends on the taxon and how you define. In our case, morphological evidence is very limited. Coat coloration or things like that really doesn't say much about Galagus in first place. And even when we do skeletal morphology, there's very little variation between those different species. But for instance, there are other morphological traits that I haven't had the chance to talk about that are significant. And this is, for instance, the shape and morphology of their penises, because they actually have very specific spines and structure of their penile morphology that seems to be diagnostic. And so again, I don't think there is a magic trait, I don't think that not just for cryptid species in general, I don't think there is a magic trait to define species and draw a line. But I think it's important when you're looking at a species to also recognize what is their sensory system and how they recognize each other and what kind of channels they're using, because that's really important for them. And for Galagos, acoustics plays a major role. And so that's why we're focusing on that in particular compared to like visual components.
Thank you very much, Dr. Pozzi.
Thank you so much. Sorry for running late.
It's okay. It's okay. It was very good. We had a lot of questions.
Thank you.
So thank you everybody for coming. The next talk is pretty much already happening. It's from Dr. I think Santiago Catalano. That's it. Thank you very much for coming.